Okay. Okay. It's 9 o'clock. Okay. We have 55. Well, okay. Good morning to all. Welcome everybody to this special seminary. Today we have a participant of the Dr. Andrew Millard. Dr. Andrew Millard is a lecturer in bacteriophage bioinformatics, studied in the University of Plymouth, gaining a degree in micro and cell biology before moving to the University of Warwick, who completed his PhD in the molecular biology of marine cyanophages. We are led the genome sequences of the marine cyanophages SPM2, following her PhD postdocs with Professor N.Man and D. Scallan in the area of marine microbiology. In 2013, moved to the School of Life Science of Warwick Medical School to take a position as senior research fellow at setup of on-ground group in the newly formed microbiology and infecting. In this moment, moving to the Leicester. Leicester is correct, Andrew? Leicester. Position as a lecturer in bacteriophage bioinformatics. Please remember question by chat or raise hand, wherever you want. And Andrew, thank you very much for this talk, and I'm giving to you the microphone and camera.
Okay, thank you for the invite. So can people see my screen now?
Yeah, we can see.
Excellent. Okay, so thank you for the invite. So today I'm going to be talking about the vast diversity of vibrio prophages. I think this summarizes what we know about bacteriophages. So we really know just the tip of the iceberg of bacteriophage diversity and what bacteriophages do. And there's many, many things hidden underneath that we still got to discover. So today I'll be talking a little bit about background of why we study marine phages, what we know about vibrios and phage diversity, and then some of the work that we've been doing and looking at vibrio prophages, their detection in metagenomes, some of the functional genes that they carry, and some of the experimental confirmation of these genes, so antibiotic resistance genes, and then if there's time, a little bit about a novel lytic vibriophage that we've isolated and found to be quite unusual. So why study marine viruses? Well, this slide shows what happens if you take a seawater sample and you simply stain it with cyber green and look under a microscope and look at the fluorescence. So these big blobs aren't stars in the solar system. These are bacteria, and these very small, tiny dots, these are viruses. So viruses are extremely abundant. There's over 1 million viruses in every teaspoon of seawater. And in fact, they're the most abundant biological entities in the biosphere, with an estimated 10 to the 31 viruses found across the globe. They form a part of the dark matter, and by this we mean the vast majority of these things are completely unknown and we have no idea of their function. The majority of them have never been isolated or sequenced, and to put this into some context, it's estimated 20% of nucleotides on Earth are of viral origin. Yet, however, if we go to GenBank or NCBI or any other large database, only about 1% of nucleotides in that database will have come from viruses. So they're vastly underrepresented in databases. To look at this another way, a large-scale study by Jennifer Brum and Matt Sullivan's lab that looked at viruses in the oceans, they estimated there was about 5,500 pelagic viral populations in the ocean. So what do they mean by a population? If we put this in a taxonomic framework, this would be a viral population will be roughly equivalent to a genera. So there's 5,500 genera in the oceans when they did this in 2016. When this was updated in late 2019, this is well over 11,000 different genera of viral populations that we could identify, but we only have sequence representative for about 30 of these, and that's just at the genus level. So when you go down to the species and the strain level of viruses, there's a huge number, and we simply don't know anything about them. So when I moved into the med school from life sciences, one of the things in a med school, well, it's quite hard to justify studying non-pathogenic organisms in a med school. So maintaining my interest in vibriophages, in marine organisms, I started to look at vibrio. So why are vibrio important? So a lot of you already know this, given the institute that you work at. So there's a huge range of vibrios. They can cause ear infections in humans. They could cause gastrointestinal disease, parahemolyticus, mimicus, and cholera in humans. They can also cause horrid, nasty skin infections, alginolyticus and vulcanificus, causing your skin to boil and bubble up. Also, a major problem in the aquaculture industry, so Vibrio splendidus, harveyi, alginolyticus, parahemolyticus again in shrimps, so they can have devastating effects on shrimp farms. And for this reason, we started to look at vibrios. And you can see, so this is just some data that's easily accessible, the number of vibrio infections. So you can get outbreaks of food poisoning, mainly by the consumption of seafood in the US. There was still a large number. The rise in vibrio cholera is thought to be linked to climate change. So as we go forward, understanding vibriophages and in particular CTX and how that might change with climate change is important, and also virulence genes. And of course, most people have probably heard of vibrio cholera. So this is estimated that there's between 1.3 and 4 million cases of cholera per year. And cholera, the toxin genes are actually carried on the prophages found within the genome of the bacteria. And it's these genes that make it such a deadly killer. So what else is in there in other vibrios that may be coming that we don't know about yet? And also importantly in the environment, vibrios are really important in cycling of carbon in pelagic oceans. So one of the things that's recently been found is that vibrio species can account for up to 20% of the microbial community when they bloom. So where there's an import of iron from Saharan dust as it blows across, this causes massive blooms in vibrio. And again, we don't know what's happening with these vibrio in terms of their prophages. So what we do know about vibrios is that vibrio cholera is known to be a large global killer, and this, as I've already mentioned, is because the filamentous phage CTX encodes two genes, CTX A and B. There's other toxigenic conversion phages known as TLC and satellite phages. The vibrio harveyi, VHML, is a toxigenic converting phage, so it makes the host more toxic. In vibrio coralliticus, the prophages within that encode toxins and are implicated in the bleaching of coral. So this is just a few examples that we know within vibrio. So what we wanted to look at is other vibrio. So get some kind of context of what we know about bacteriophage genomes. It's very little. So this is just the number of bacteriophage genomes that have ever been sequenced up until 2019. We can update to 2020, but the numbers don't really change that much. So since the first phage genome was isolated and sequenced in 1980, 65 and submitted in GenBank when it started, so it was actually sequenced in 77. We're now about 10,000, and in 2020 we're somewhere up here about 16,000 phage genomes have ever been sequenced. But if we put this in some context, so 16,000 phage genomes, but there's greater than 200,000 salmonella genomes alone. So when we look at the genomes of phages, we know very little about them because they're massively undersampled. And not only are they undersampled, they're also biased towards a particular group of organisms. So this is just a breakdown of the different phages. So nearly 16% of all phages that have ever been sequenced have been isolated on Mycobacterium smegmatis, and this is due to a program in the US known as the C-phages program. And then we have other common things, Escherichia, Pseudomonas, and other bugs that you might expect. Vibrio actually pops up here around about 5%. When we started this work, it was far less, and the reason for this is this paper that identified a major lineage of new non-tailed DNA viruses where they sequenced several hundred, that brought it up from a very low number to about 5% of all phages ever sequenced affect Vibrio. But not to be deterred, we continued with the work I'm about to describe to you. So this is what we know about bacteriophages at the start of the project. So this is a tree, a phage proteomic tree, and this is the total diversity of phages infecting Vibrio that we knew about when we started this work. So one of the things that you can notice is that there's small lines coming off, and these are just indicative of taxa. So these are defined taxa of different vibriophages. And one thing that stands out straight away is that most phages that have been isolated have no known taxa. And this holds all true for most phages. So the ICTV, which classifies them, there's a huge amount of work going on in the background to try and give all phages classification. But in the case of Vibrios, most of them don't have any taxa. Some of the things that stand out are Phi-CTX, which is over here. This is very, very distantly related to another phage that is quite well known that infects Vibrio, which is KVP40. There's a huge amount of diversity. So the aims of this work, so most of this work was done with a very talented PhD student of mine called Slav. So his first aim was to determine the diversity of prophages found across the genus Vibrio, and then go on to have a look and see if similar phages are found in different Vibrio species. And when he'd done that, was to identify auxiliary metabolic genes in Vibrio phages. So what are auxiliary metabolic genes? So these are genes that add some kind of function to its host. So a toxin gene would be known as an auxiliary metabolic gene. Antibiotic resistance genes, so if the phage carries the resistance gene, does it then confer this resistance to the host? That would be classified as an auxiliary metabolic gene. But there's many, many other processes for the that might be considered within AMGs. For this talk, I'm just going to mention antibiotic resistance genes. We actually found a lot of other AMGs. So just to be clear, I'm going to be talking about two different groups of phages. So I'm going to be talking mainly about prophages and a little bit about lytic phages. So the common pathway for a prophage is that temperate phage, which can become a prophage, is that the phage is floating about. It attaches to a bacterial cell, it injects its DNA, and then this DNA gets incorporated into the bacterial chromosome. This can then divide, so you get two daughter cells, each of which contains a prophage incorporated. At some point in the future, it may never happen, it can go several hundred generations, this prophage gets induced, it starts to replicate, and you produce more virions, and then they get released. But this stable prophage state is what we're mainly going to be talking about in the rest of this talk. And this is what we wanted to investigate. So how did we do it? So we took an approach of going to the databases and finding every single Vibrio genome that had ever been submitted to GenBank. We also went to the short read archive and pulled out all reads of vibrios that had never been assembled. We used a database infrastructure called Entrabase, which has a huge amount of QC of the data. So we trim the reads, check they are vibrios, and use a number of quality control things for assembling genomes. So we initially assembled over 9,000 genomes. We then use something known as an API for FASTA, which predicts the presence of prophages within those genomes. We do gene annotation and calling. We do some curation. We then, for the purpose of this talk, concentrated on looking for antibiotic resistance genes with a program called Abricate. Once we found those genes, we went on to look at the function. So in the end, after quality control and removal of some genomes, we ended up with a final data set of just over 8,500 vibrio genomes. And these were representative of 110 different vibrio species, with 144 of which had unknown or undetermined. So we didn't try and give them any species, we just accepted that they weren't. Under our quality control procedure, we also checked that if it said it was Vibrio cholera, it was actually most likely a Vibrio cholera and not a Vibrio parahemolyticus, but this was only done for known species. We didn't make any new ones. And then we're able to identify prophage elements within these genomes. And we found over 17,000 prophage elements in the 8,500 Vibrio genomes. So most Vibrios have at least one intact prophage within them. And as you can see here from the distribution, most of them had at least one, but we found some genomes that actually had 6 prophages within them. So the next thing we did was to look at these prophages and work out if they were similar to anything that was already known. And what this is here is the number of genomes, and these are known phage genomes down the side. And we used a 95% average nucleotide identity cutoff determined by something called MASH to say whether or not they were the same thing. So generally speaking, in phage taxonomy, if something has greater than 95% ANI, then it's the same species. And what stands out really clearly is there's a huge number of phages that were found to be similar to Vibrio CTX5, so the cholera causing one. And this didn't really surprise us, as our data set was dominated by Vibrio cholera genomes. What we can see, we find a number of other phages that are already known. So these are integrated in the genomes that we were looking at and are similar to known phages. What was of interest is this big region down here. So this is the just over 2,200 genomes that were unclassified. So are all these genomes exactly the same thing, or are there many, many genomes that are unclassified? And another thing that we found is that we found Escherichia virus 5X174. So we're not saying that Vibrio cholera contains 5X174. What we're saying is, despite our best efforts at cleaning up the genomes, some people submit the genomes and don't remove the spike in DNA of 5X174 when they assemble their genomes, and it gets caught up, and then we identified it. So we're not saying it polyphages are found to infect Vibrio. So as I've said, lots of phages are similar to CTX, and it was these 2,500 phages that aren't similar to anything in GenBank. And what we could actually do is reduce the redundancy in this data set, and we found 556 representative phage genomes that are extremely diverse. So these 2,200 genomes aren't all the same thing. There's actually 556 different groups of phages within the Vibrios that we started with. So we then went on to look at the diversity of these through a phage proteomic tree. And to do this, we use a BLAST P approach using something known as VipTree, which is quite common and used to look at bacteriophage genomes. And what we've done here is plotted this. So this is a tree of the diversity of different phages. So every single faint little line represents a different phage genome. And then we've added a little bit of colouring on top. So the red means if you find red at the end of the tips, that phage genome was found in Vibrio cholera, Parahemolyticus is in green, and Vibrio vulcanificus is in yellow. We added a colour for all the different genomes, but I only highlighted some of the most important ones. When there's no colour here, white genome, that's representative of where the phage is already existing in a database. So if it's white there, we know it already exists, and all the ones with colour are new things that we've found. So we can see we've got a huge diversity of new phages. One of the things that jumps out straight away is that all these phages here, these are innoviridae, so these are phi-CTX. So if we remove that, we look at exactly the same data, just with phi-CTX removed from it because there were so many of them. Same thing applies. If it's white, we already knew that there was already a representative of that phage, and if there's any other colour, all of these phages have been found as prophages, but never been found in the database before. So it looks like there's a huge diversity there. So another way of looking at this and grouping phages to reduce some kind of redundancy within the data that we have is to use a program called vContact2. And this gives you a network overview of how the phages group together. So hopefully you can see these like clusters of balls that are close to each other, and then we've overlaid colour on the top of these. So the red, these dots that stick about every now and again, this is a Vibrio phage that was known at the start of this study, and the black is any other phage that was known at the start of this study. And then the lilac-y bluey colour, these are all new phages that we identified within this study. And what this allows you to do is start to see clusters. So you can see discrete clusters of phages that are grouped together. And this allows you to apply some taxonomy on the top of them, and you can define what is known as a viral cluster. And these roughly equate to a genus. So there's a huge number of probable new genera that we found by looking at prophages that weren't known at the start of the project. So the next question is, are these phages real? So with any kind of prediction, it is only a prediction. So ideally what we'd like to do is take every single genome, try and induce the prophages and show that you can get a functional phage or virion at the end of it. Only with data of this scale, you can't do 8,500 inductions. But what we can do is take existing data from virome data sets. So what was done here is people have taken previously produced viromes. So a virome is where people have gone to the environment, they filtered out all the bacterial from a seawater sample, and they're just left with and enriched for all the viruses that are present. And then they sequence this. And this was done as part of a large project called TARA, and each of the dashes at the bottom represents a different sampling point as part of a large global survey to look at viruses in general. So we then took this data and we had 1 to 556 different different prophages. So each dash along here represents a different prophage, and we mapped all of this data against our prophages. And if our predicted prophage was present in a viral sample, we could look at the genome coverage. The coverage hasn't been normalized in any way to the size of the database of the viromes, which varied quite a lot. But what this does tell us is every time we see a little dot here, this tells us the rough genome coverage of read mapping against our representative genome. So we can see for this particular representative genome that we find it in a number of different samples. So this suggests that this particular prophage was found in a large number of different viromes, and therefore it is producing a actual virion. This particular virome here, where there's lots and lots and lots of red dots, this is because I think this is one of their testing samples when they were optimizing their procedure, and they sequenced this sample to its coverage far higher than they did for any other sample. So this allows us to detect things. So of our 556 representative genomes that we predicted, we could confirm that 188 of them are actually present in the environment and do produce actual virions based on read mapping. And this was using standard cutoffs. So it looks like a lot of our predictions do hold true, and you can probably go out in the environment and find these prophages. What they're doing out there, we still don't know. So we also did some further lab work on a small scale. So we were able to take some representative strains of vibrios that we have, so Vibrio parahemolyticus and Algae nolyticus. So we sequence them with long read sequencing so we can get complete genomes, so chromosome 1 and chromosome 2, plus the plasmids that vibrios carry, and this one just had 2 chromosomes. And we are able to identify the prophages on both of these, and then we were able to induce prophages that produce a virion. So this is the viral particle, so we get different morphologies, and sometimes we get long filamentous ones, and in this one it was a little bit more clear cut. So we have one prophage that was induced, we get a really clear image of the virion, and we also able to sequence the virion particle and confirm and show exactly where it inserts. So we did some experimental work to confirm just a small number, so we've only confirmed 3 within the lab. But all 3 of these represent new species of vibriophages that hadn't previously been done before. So we've also done some work with some other groups. So this work was driven by Julia Frunz's group with Eugene Pfeiffer in Germany. So they'd been working on 2 phages known as VNP1 and VNP2. And these are the 2 phages. They'd noticed in their sequencing they got higher coverage, suggesting they were induced, and they were able to identify the phage particles and predict where they may occur in the genome. And then they came to us and asked us whether or not they could find these in other vibrios. So some of the work that they came out was they were able to label their phages with a cherry red marker and actually monitor the production of the phages. So this video just shows their vibrios going, and then you start to see the red producing as they can see the phage starting to pop out because they stuck this cherry red label on. So this was a really cool video of theirs that that was involved in their paper, but they came to us because they wanted to know whether or not VNP1 and VNP2 were found in other vibrios other than the single strain of vibrio nitrogens that they looked at. And the answer to this was yes, we could show that vibriophage VNP1, VNP2 was actually present in a range of other vibrios, which is really quite important because what they were finding is that although they could knock out these prophages in the original host, and it didn't cause any change in growth rate, which is the data they presented here. The prophage-free strain exhibited a higher tolerance towards DNA damage and hypoosmotic stress in the free form. So it actually shows that carrying prophages actually has an effect on the vibrio nitrogens. There's a little bit more evidence that some of the prophage predictions we have are actually real phages. So we then went back to look at one of our original questions and try and work out whether or not different prophages are found in the different species of vibrio. And what we are able to confirm is, so we're now looking at just these vibrio clusters, which we've got from V-Contact. So vibrio cluster 515 is present in a huge number of samples, and these are all vibrio cholera. So we didn't find it in any other vibrio species, and this confirmed previous results. So this cluster is also known as phage 919TP in the literature. We're also able to go on and show that phage CTX is not limited to Vibrio cholera, and we can find it in Vibrio mimicus, which has been reported, but we expanded the range of Vibrio mimicus, or the number of strains that it can be found in. Some phages like H20-like, which are found in Vibrio anguillarium, we confirm this, and again went on to show it's found in more than just, it's limited to just Vibrio anguillarium, but there's a large number of them. Whereas other groups of phages, such as cluster 358, this is quite promiscuous. So this isn't isolated in a single Vibrio species, but is found in a larger number. So Parahemolyticus and Algelinolyticus. So a number of them are found. So this figure here is the VC cluster number. So each one of these just represents a group, and this is the number of different Vibrio species that we found it in. So this one is quite unusual. So it's found in up to 16 different Vibrio species, suggesting it can move about from one host to the other, whereas a lot of them were restricted to just one. Okay, so this is T31, which is the promiscuous one. So the other question that we had at the start of this was antibiotic resistance genes. Do prophages carry antibiotic resistance genes? So this is something many people are interested in. Are they major vectors of moving them about? So we went through all our prophage predictions and started looking for antibiotic resistance genes, and we can find antibiotic resistance genes. Somewhere around about 1.2% of prophages carry these resistance genes, and they are found across a range of different vibrios, including cholera and Parahemolyticus, as well as Splendidus. And the number of genes in the different antibiotic classes varied. There wasn't a particular type of gene that was found in any particular type of class. So although they are there, does it mean that they are important? Are they doing anything in the environment? Are they doing anything, or are they even functional? So what Slav went on to do is to look at the function of these by using a heterologous expression system. So one particular gene that we're looking at is a CAT gene that confers resistance to chloramphenicol through chloramphenicol acetyltransferase. So this is the phage genome. It clearly fits within this phage genome, definitely phage-like. And what Slav did was to actually test a range of different genes. So from the predictions he had of different antibiotic resistance genes, he took each of these individually and he expressed them in a plasmid that we produced in E. coli. So we fitted each one of these into this particular region and then looked at constitutive expression of them. So we have a control here. So this is our control plasmid. Down the bottom here, thrown in antibiotics, and you see you don't get any growth. This is a control with no chloramphenicol, and it grows quite happily. And then if you add in the plasmid with the chloramphenicol gene in, you can see that it also grows quite happily, showing that it confers resistance. And we've gone on to look at the minimal inhibitory concentration of these. But he was actually able to show that prophage encoded genes conferred resistance to at least 6 different antibiotics. So in terms of the prophage summary that we have, we're able to identify 125 viral clusters of prophages that infect Vibrio, many of them which had never been seen before. We are able to identify many of these can probably produce an actual virion based on the Tar Oceans data set, and that they encode antibiotic resistance. And the last 6 or 7 minutes of my talk, I'm just going to mention some of the novel lytic phages that we've been looking at. The lytic phages are free form, and one of these novel lytic phages that Slav had also isolated is known as SM033. So it produces quite nice images, and if you look at this fuzziness isn't the image being out of focus. It looks like these phages have an unusual structure, and it appears they have like almost hair-like structures appearing on their tail. So this phage was quite interesting to look at under the TEM. And Slav went on to sequence this phage and get the whole genome sequence, and it's also quite interesting in terms of its genomic architecture. So most phages tend to be smaller than 200 kb, but this one is about 320 kb, so it's a huge genome, and it contains a huge number of genes that we have no annotation for or no known function. This isn't too unusual with bacteriophages, as this often occurs, but this one was unusual in simply the size of it and the sheer number of hypothetical genes. So we've gone on to look at this in a little bit more detail. So as I've said, the genome is about 320 kb in size. And interestingly, it doesn't carry any tRNAs, which is an unusual feature for large phage genomes, which tend to carry a lot of them. Large number of genes, 280 of them hypothetical or unknown function, so a very high percentage. It was also quite interesting in the phage was able to cross-infect a large number of different vibrios across a range of different species. So these are just representatives of the different species of vibrios that it can infect. So it wasn't restrained to a particular vibrio host. So we went on to study in some more detail and a look at the proteomics of this phage. So by doing large-scale proteomics with an Orbitrap, we're able to identify 88 proteins that are associated with the virion. So you purify the phages and then carry out mass spec on them, and this tells you which genes are associated with that virion particle. So 20 of them were structural proteins that we predicted based on bioinformatics, which is nice. So we were able to confirm 20 of them. And then some other interesting things we found, which is known in some other large phages. So they encode subunits of an RNA polymerase, and they package these within the head of the phage particle. There were several subunits that are packaged within the head that presumably used immediately once they're inserted into the bacteria when it infects. And more importantly, I guess, or also of interest, is that we're able to identify another 60 new structural proteins associated with this phage, none of which had been annotated as structural proteins in any other phage that's currently out there. So it expands the database. To give some idea of how different this phage is, we've done a similar thing with clustering of B contact, and it forms a group down here. So it's distantly related to this group of phages here, which are large phages, but it kind of stands out on its own down here. If we do phylogenetics on this, the phage of interest that we're looking at is here. So Vibrio phage VB SM033. So this is built on a terminase tree. Yep. So it's similar to one other phage, and that's Vibrio phage Benashii. So on the tree, these two things appear to be very, very close. When you take the genome sequence, if you were to do a blast N of these two sequences, they would show very, very little similarity. So although they're close on their tree, they're actually really quite different. And they likely represent a new family of phages or a subfamily of phages compared to what's already known that's out there. So they're really quite different. So given this phage had such a big genome, what we wanted to know was, can this phage be involved in generalized transduction? So this is where the phage mispackages host DNA in its head and then move this from one thing to another. And if it could, being able to move 320 kb of host DNA, it may have an important role in gene transfer, especially of antibiotic resistance genes. So what we did is set up a qPCR assay to try and quantify this. So the problem with doing this is any phage, when you purify it, you have your phages and they have their host DNA, they have their own DNA packaged inside them here by the blue, and then they can occasionally package green or host DNA. But when you purify them, you always get free DNA floating about however you purify them. So if you want to quantify this, you need to make sure you remove this. So we went through the process of making a qPCR assay But because the phage DNA is packaged within its head, you can use the DNA as treatment, and that removes the free DNA. And then again, you can use qPCR to check what is there and quantify exactly how much, which is what we did. Then you disrupt the heads of these phages, and that releases the DNA from within them. And then we can check to see if there's any host DNA within them via qPCR and quantify a level within it. So we were intrigued to know whether or not this large phage could carry out generalized transduction. And the answer was that it couldn't. So although Slav spent several more months working on this, we couldn't detect any signs of generalized transduction in this quite large phage. I must say this was at our limits of detection, so it was occurring below our limits of detection. So in summary, we looked at both vibrio prophages and a single large vibrio phage. So the prophages, the large majority of them have never been seen before. We're able to confirm some of these by a number of different methods. The prophages carry many genes that can alter host function. We just focused on ARGs for this talk, but we found lots of other genes that I'm happy to talk about. So they can encode antibiotic resistance genes, and they can be functional in a heterologous host. We also found a novel large vibrio phage that represents an understudied group, and in this case it was unable to undergo or carry out generalized transduction. So this work has mainly been carried out by this guy here, so Slav. So unlike me, who happened to lose my hair as his PhD went on, Slav gained more and more hair. So this is my group as it was at Warwick University, and about halfway through Slav's project, I moved to the University of Leicester. So I just need to thank my long-term collaborator, Dave Scanlon at the University of Warwick, and Luigi from the University of Geneva, who provided a large number of strains in this, and everyone else in my group, and the people for the funding. And thank you for your attention.
Thank you very much, Andrew. Very interesting, your talk. Gentlemen, ladies, questions? Ready, Dr. Ricardo?
Hi, thank you very much for your very interesting presentation. It's awesome, such a diversity that, and it's very well understudied. Because we are in the aquaculture academy here, I would like to ask you whether or not you have any experience applying this kind of work or some applications for aquaculture. As you showed at the beginning, there's very much concern for stream aquaculture and oyster aquaculture about the problems carried out by this kind of stuff. So can you
so applying phage therapy in an aquaculture environment?
Yes, or whatever you think this information can be applied.
So I think in an aquaculture environment, knowing what vibrios you have and what genes they contain is quite important in terms of what might be coming next. So what I didn't touch on today that we've also studied is the effect of temperature. So temperature is known to induce prophages, and we have some evidence in these vibrios that as the temperature increases, the number of prophages get induced, and then these release into the environment. So if these prophages are carrying antibiotic resistance genes or toxin genes, it means that with an increase in temperature, you may get more death of whatever the aquaculture fish, shrimp might be, because they're spread into other vibrios. And that's one of the things we're interested in, if these different environmental cues have an effect on the spread of disease because the phages are moving from one vibrio to the other. So we're definitely not there yet, but that's what we're interested in looking at further. because as we can see, some prophages are found in lots and lots of different vibrio species. So it suggests they can move from one thing to the other. And if any one of them was to acquire a virulence gene, it suggests that some commensal vibrios could then become virulent, and this might present an issue. In terms of phage therapy, the only phage therapy I've done in aquaculture is vaguely with Eduardo, and he's driven all that. So we've looked at phage therapy in other environments, but I don't see why phage therapy wouldn't work in aquaculture. And some of the large phages were isolating that can affect a range of different vibrio species, would definitely be useful tools because they have quite a broad host range. And this is quite important when lots of vibrios cause disease. Instead of having to get 10 phages, could have one phage that could target a number.
Thank you.
Does that answer your question?
Thank you. Emmanuel?
Hi. Well, thank you for your presentation. It was really interesting for me. So I really like the importance that you gave to the antibiotic resistance that these prophages could induce in the vibrio bacteria. So I have two questions. I think you kind of already answered those with Dr. Ricardo's question. But is there a possibility that these prophages are enhancing the virulence or the severity of the infections that these bacteria could induce in humans? And is there a way in which we can use these prophages to develop a treatment or vaccines against it in humans?
So yes, they're definitely causing disease in humans. So cholera is the classic example. So it's known that cholera is caused by the CTXA and CTXB toxins, and that's carried by a phage. And if you don't have the phage, you don't get cholera. So clearly they are. And in Parahemolyticus, the phages also carry virulence genes, and that's known. So could we use these prophages to help in phage therapy? Yes. Whether or not it's a good idea, I'm not entirely sure. So generally lytic phages are used, but what we could do is take prophages that we know to be found in Vibrio parahemolyticus, for instance, and you could engineer those so they don't have a repressor. So when they infect their host, they produce a lytic infection and cause it to die, or use them as a delivery system. So one of the issues in phage therapy is often finding a phage that will target your bacteria of interest. So with a prophage, you know it targets that bacteria because it's already present within it. So if you can engineer it not to be temperate and not make that prophage state, you could use it as a therapeutic. Whether or not that's a good idea, I don't know, but you could do it. Generally, people suggest not using prophages because there's a danger that it could revert back to something else. But we can use the information within them. So all the phages carry endolysins and lysins because they have to lyse the cell. So we know that those endolysins and lysins will target that bacteria because they have to get out, but it works both ways, and people are now being able to use endolysins and lysins in gram-negative. So if you overexpress them, you can use them, and you then know that it will target your bacteria of interest. It does take a fair bit of engineering because of the cell wall in the gram-negative, but Lone Bromsted's group in Copenhagen has done that for E. coli and Salmonella. So in theory, there's no reason it wouldn't work in Vibrio. So I think we could use that information.
Okay. Well, thank you very much.
Thank you, Manan. Good question. Good question. Okay. Juan.
Sí, ¿me pueden escuchar?
Sí, yes.
Okay, thank you for your talk, Dr. Miller. I have a question according to the phylogenetic trees. In the phage SM33, the amino acid sequence of the major capsid protein was used to construct the phylogenetic trees. But it's normally seen in literature that to use both capsid and DNA polymerase as biomarkers. So shall we report both even if they show the same results?
Should you report? So we actually used a number of different genes. So in the tree, I did use the major capsid protein that I presented, but we've done it with the terminase gene and also a DNA polymerase. But our DNA polymerase is actually not a great marker in this one because it has an intene in the middle of it, which will confuse the phylogenetic signal. So which gene that you should use probably has to be interpreted with each bacteriophage that you're looking at. So not every marker gene will be suitable for every single bacteriophage. And if they show the same things, that's encouraging. That's what you want. Ideally, in this study, we would have liked to have used a cord protein tree, but these phages are so different, there isn't a core set to use. So that's why we'd only gone for a smaller number. So the V contact uses like a larger number to cluster them as well. So to answer your question, if they produce the same thing, yes, I'd still present both trees because that's encouraging. You'd expect marker genes to produce the same phylogeny, and if they didn't, you'd want to know the reason that they didn't.
Thank you. That answered my question. And I have another one, if Dr. Quiroz allows me. Well, there are some bioinformatics tools used to predict the host that the phage may affect. Well, in your experience, how useful it could be used, these tools, before we proceed with experimental analysis, for example?
It depends on what level you're talking about in terms of host prediction. So I think you have to remember it's always going to be a prediction. So there's different approaches. So things that predict hosts based on CRISPRs can be quite useful. I don't believe that you'll get a host prediction at a strain level for a bacteria that's accurate, or even a species at the genus. Then yes, I think that's a good predictor. But if you're talking about strain level prediction, I don't particularly believe that. I mean, lots of other people that I work with wouldn't believe that either, but they might believe you at the genus level. And as you go up the higher taxonomic levels, the accuracy tends to increase, which is a good thing, but also is often less useful if you can say it infects a particular kingdom, Peter Cannon, order of bacteria. How useful is that as a tool if it only predicts accurately at the order? So I'd say interpret them with caution and possibly look for more evidence. It depends what your question is. They're a good starting point to get overviews, but I wouldn't draw firm conclusions on them without some kind of backup.
Thank you. And the last one. Well, I have seen a little more in Pseudomonas phages, for example. How could we study from a bioinformatics perspective the relationship between phage bacteria in terms of bacteria defense, for example, in CRISPRs?
Sorry, say that again. I didn't quite catch the start of the question.
Yeah. What I say is I have seen a little more in Pseudomonas phages, but how could we study from a bioinformatics perspective the relationship between the phage bacteria in terms of bacteria defense?
How easy is it to study, or how cool?
Yeah, how cool.
I mean, I think it's a really interesting, I think CRISPR and understanding CRISPR and anti-CRISPR proteins and anti-CRISPR proteins as they now found in bacteria, I think that's a really interesting thing to do. Bioinformatically, I mean, there's lots of tools out there that will predict CRISPR arrays and CRISPR proteins, so all of those things can be done, and I'm a big fan of that. But even as somebody who spends a large amount of time doing bioinformatics, I think you need to go back into the lab and confirm whatever prediction you have is for some of the data. So I think it's an interesting area to study for sure.
Yeah, thank you.
Thank you, Juan. Dr. Anna?
Hi.
Hello.
Sorry if I don't put my camera, but I am not in conditions for you to look at me. Okay.
Yeah.
And following the Emmanuel's question, I am very interested in antibiotic-resistant genes, and there are many researchers paying attention in bacteria to study this. Do you think we should pay more attention in phages instead?
I think it will depend on the bacteria that you look at. So I'm interested in antibiotic resistance genes and phages in particular, and whether or not they play a role. So there's, in the phage-like area, I guess there's lots of reports, and often they contradict. So the two largest studies to date, one of them says that antibiotic resistance genes are rarely encoded in bacteriophages. And then another more recent paper that came out 12 months later in the same journal said antibiotics genes are often carried in bacteriophages. So I think we still don't know enough, and it will depend on the system. So in vibrios, we think they don't have that big a role. So 1.2% is a fairly small number of phages carry resistance genes, but we've done a similar study in E. coli, which tends to have far more prophages, and in them on a smaller subset. So in E. coli there's 150,000 genomes, but we've only looked at 5,000, and I think it was closest to 15% of the prophages were carrying an antibiotic resistance gene. And when you get into that kind of level, I think they're probably important in moving them about and having a role. And other things that people have looked at in terms of antibiotic resistance and phages is they've noticed in, what did they do in Pseudomonas, that there's no known antibiotic resistance genes in the prophage, but if you remove the prophage, you also increase the sensitivity of the bacteria to antibiotics. So there's unknown genes in there, and they're all hypotheticals at the moment that are conferring resistance, but we don't call them antibiotic resistance genes at the moment because we don't know what they do. And I suspect, given the large number of hypothetical genes within prophages, there's probably many more instances of that. So I said, yeah, you should probably pay more attention, but I couldn't give a definitive they're more important than plasmids or any other thing. I think they're often overlooked, and more research is needed to work out if they are important or not.
Yeah, and do you know if is there any correlation?
Correlation between
antibiotic resistant genes from phages and bacteria?
Do you mean, do they get them from the bacteria or?
Yes.
Yes, we think so. It looks like it. So in the vibrios we've looked at, the genes that we found conferred resistance, there was also a copy in the bacterial chromosome as well. So they'd have two copies of a TEM gene, or they'd have two copies of, what else were we looking at? The CAP gene, for instance. So there was multiple copies of the CAP gene present within the vibrio, and one was in the prophage and one was in the bacterial chromosome.
Okay, thank you very much. Very interesting.
Thank you.
Thank you, Doctor. Any question? Any more? No. Let me see. Nope. Let me see the chat. Yes. Carlos. Carlos, please. Hello.
Hello. Good morning, Andrew. Good morning, everyone. I have a little question for Andrew. My question is, you talk about the prophages and the role with their host, and you search for prophages in your metagenomic analysis. So my question is about the lytic phages. Do you do any test with them, and do you look search lytic phages in the environment? I don't know.
So after meeting you several years ago at a conference, I think I mentioned we were looking for lytic phages, and you told me how easy it was to find lytic phages in your shrimp ponds, and then so we did look for these lytic phages. So Slav spent 18 months looking for lytic phages, and he found none whatsoever in hundreds and hundreds of samples that we've collected from everywhere. And then he, well, we don't really know what changed. He just tried some more strains and the same water samples, and then we're able to get about 100 phages. So we have about 100 lytic phages that we've sequenced, but we haven't finished the analysis of yet. So Slav had this very hard experience with not being able to get anything, and then suddenly finding that he could, but it really depended on the host that he was using. So we had about 250 hosts in our collection, and it was only on a small number that we could get any phages from. So we were getting to the point of giving up and about to stop because we couldn't find any lytic vibrio phages, and we were reading all these reports that said, oh, you just isolate them, it's really easy. And then we spoke to you, and you kindly sent us some samples and things as controls. So yeah, we did try that, and we did find we can get lytic phages, but it was dependent on the host we used, but we don't know why.
Yeah, that's what I expected. Okay, I don't have any more questions. Thank you, Andrew. Excellent.
Nice to see you, Carlos.
Thanks.
I can't hear you.
Sorry. The time is over for the seminary. Gentlemen, students, thank you very much. It's, let me see one. So cool, see you. Okay, so cool, see you to talking. Okay, Andrew, thank you very much. It's very important and interesting, the study of bacteriophage. Okay, and so thank you for your time. Thank you for your speech, and wherever we have contact for the work pendant, the papers.
No, thank you for the opportunity.
Thank you, and see you with another time.
Okay, thank you for the opportunity.
Dr. Ricardo, something say?
No, thank you very much for your time, Andrew. It was great.
No, thank you.
Okay. See you, Andrew.
See you.
Bye-bye.
Be careful.
You too.